Welcome back to our Week 5 B-cell module for Applied Immunology. This is Lecture 5B, where we will learn about the signaling steps involved in the activation of mature naive B-cells following their successful development. Today we will discuss signal transduction pathways that occur downstream of the B-cell receptor, and how these allow B-cells to assume an activated cellular state. We will also go over two categories of antigens that are recognized by the BCR, which are Thymus-dependent and Thymus-independent antigens. And we will learn how these differ with respect to a process called T-cell help that's offered by follicular helper T-cells. We will then end this lecture with an overview of how T-cell help leads to germinal center formation, which can specialize antibodies through the processes of somatic hypermutation and affinity maturation, as well as class switching. This talk will lead nicely into the remaining lecture for Week 5, where we will learn about the functions of B cells, as well as the antibodies that they produce. To start off today, the signal transduction portion of this lecture won't be nearly as intensive as the Week 4 lecture on T-cell signaling. This is because the BCR and the TCR share a lot of similarities in their signal transduction pathways, so we've already learned about a lot of these. We've established that the TCR can signal through cytoplasmic ITAM domains that are present on itself as well as the co-receptor CD3, and these ITAMs are shown in this diagram in yellow. Now these become phosphorylated and then kick off a signaling cascade that involves numerous components of signal transduction. These include other kinases that phosphorylate target proteins, adapter and scaffold proteins that recruit signaling members together, phospholipases and lipid kinases that manipulate the status of membrane-associated lipids, phosphatases, which again remove phosphate groups from target proteins, and lastly, small g proteins that act as on-off switches for signal transduction. All of these signaling components ultimately function by inducing the activation of certain transcription factors, as well as by modulating other components of T-cell biology, which include cytoskeletal structure, adhesion to extracellular ligands, and metabolic reprogramming. B cells signal similarly to T cells in that their membrane-bound BCR exhibits antigen specificity that is then translated into intracellular signals through the phosphorylation of cytoplasmic ITAM domains. The BCR does not possess its own ITAMs, but it associates with the transmembrane proteins Ig-beta and Ig-alpha. These are invariant proteins that are associated with the immunoglobulin heavy chain and can be targeted by tyrosine kinases. Although the specific proteins involved with BCR signal transduction may vary from TCR signaling at various points in these pathways, BCR signaling acts through these same types of signal transduction components to ultimately assume an activated cellular state. And this is similarly defined through transcriptional, cytoskeletal, adhesive, and metabolic changes to the B cell. B cell signaling starts with a process called BCR cross-linking, which we will briefly reference in the last lecture. Cross-linking occurs when multiple BCRs are brought in close proximity to one another, and this is done by a multivalent antigen, which again is an antigen that has multiple binding sites for the same BCR specificity. Cross-linking of multiple BCR molecules is required for BCR signaling, which explains why weak affinity or monovalent antigens that are encountered during development do not stimulate the BCR, and this leads to the generation of clonally ignorant B cell clones. Once cross-linking occurs, the ITAMs of multiple Ig-alpha and Ig-beta proteins are recruited together, which allows them to be targeted by the first set of tyrosine kinases in this signaling pathway. And these are called Fin, Blick, and Lin, and they're shown here in green. Note that this is a very similar initiation step like we saw in T cells, and that like the Lick kinase that initiates TCR signaling, these three kinases present in B cells are also Src family kinases. Phosphorylation of two tyrosines on each ITAM on Ig-alpha and Ig-beta allow for the recruitment of another tyrosine kinase called Syk. And this is because Syk has an SH2 domain, which again recognizes double tyrosine phosphorylation motifs. Syk becomes activated simply upon binding to the phosphorylated ITAMs. And Syk functions very similarly to ZAP70 that we saw in T-cell signaling, and that both of these proteins are recruited to phosphorylated ITAMs on the cytoplasmic domain of the antigen receptor signaling complex. Like ZAP70 in T cells, Syk in B cells is a kinase that is required for initiating numerous downstream signaling modules of B cell activation. B cells also have an analogous process to co-stimulation through CD28 that we saw in T cells. In B cells, this is achieved by a complex of accessory proteins that are called the B cell co-receptor complex, which consists of CD19, CD21, and CD81. CD21 binds to a form of complement C3 protein that's deposited on pathogen surfaces, so co-receptor activity occurs when the BCR is bound to an antigen on a pathogen that is also the target of complement activation. Like co-stimulation, B-cell co-receptor signaling greatly amplifies the activation signal that's relayed through the BCR, and it does this following phosphorylation of a tyrosine residue on CD19 by the Sarc family kinases Fin, Blick, or Lin, which in turn recruit PI3 kinase to generate the phospholipid PIP3 at the plasma membrane. B-cell co-receptor signaling produces PIP3 at the same time that the kinase Syk phosphorylates a scaffold protein called SLP65, which is sometimes referred to as Blink. SLP65 functions similarly to LAT and SLP76 that we saw in T cells, and that SLP65 is phosphorylated on several tyrosine residues by Syk, which in turn allows for the recruitment and assembly of multiprotein signaling complexes. Therefore, the generation of PIP3 by PI3 kinase can synergize with Syk-dependent SLP65 phosphorylation, and these events are responsible for initiating distinct modules of B-cell signaling. Now, although B-cell signaling involves some different proteins, they all have analogous functions that follow the same logic of TCR signaling and lead to essentially the same modules of signal transduction. The first module is mediated through PIP3, which recruits the kinase AKT to the plasma membrane. Like in T cells, AKT signals through another protein called mTOR, and the result is that this modulates cellular metabolism in order to allow for energy-costly functions of activated B cells. And these include the production of effector molecules like antibodies, as well as their rapid proliferation. The second module is mediated through both PIP3 and the scaffold protein SLP65, which allows the kinase BTK to phosphorylate and activate the lipase enzyme PLC gamma. This is the same PLC gamma that we learned about extensively during T-cell activation, which functions the same way by hydrolyzing PIP2 into DAG and IP3, and this leads to the same mechanisms of intracellular calcium ion influx that we saw with T cells. Similarly to TCR signaling, PLC gamma is responsible for driving changes in transcription factor expression by activated B cells, which we will discuss more shortly. However, unlike in T cells, the transcriptional program induced in activated B cells does not actually converge on the IL-2 locus. The third signaling module is also mediated through PIP3 and SLP65 and involves the VAV-dependent induction of actin polymerization through another protein that's called WASP. This allows for cytoskeletal rearrangements that are required for processes like internalizing antigen that's been bound to the BCR, and this allows the B cell to process it for presentation on MHC class II. Notably, VAV is also involved in the recruitment of small G proteins that are required for promoting the expression of integrins involved in cell adhesion, and this is essentially the same fourth module that we saw downstream of TCR signaling in T cells. Since we already extensively covered signal transduction downstream of PLC gamma activation in our week 4 lecture on T-cell signaling, we won't go into those pathways at length today. But I want to reiterate that both the BCR and TCR share very similar signal transduction pathways that link antigen recognition to cellular processes that are involved in lymphocyte activation. We will next learn about the transcription factors that are induced downstream of BCR signaling, but in order to do so, we first need to learn more about the two main categories of antigens that are recognized by BCRs and their downstream effects that these have on signal transduction. The first category of antigens are called Thymus-independent or TI antigens, and this is because they are capable of inducing antibody responses independently of helper T cells. This means that BCR signaling and subsequent antibody production occur solely through BCR and usually pattern recognition receptor signaling, so they don't require additional signals from helper T cells. TI antigens are usually polyvalent antigens, which include highly repetitive molecules such as polysaccharides that are found in bacterial cell walls. These antigens also consist of structures other than proteins. Again, these are often carbohydrates. The repetitive motifs found in polyvalent antigens can cross-link several BCRs together and enable their activation independently of the B-cell co-receptor. And the effect of this is that it induces RAS and MAP kinase signaling downstream of PIP3 generation and leads to the activation of the transcription factors AP1 and NFAT. At the same time that this occurs, bacterial PAMPs capable of triggering TLRs expressed by B cells, this includes TLR4 activation by bacterial LPS, can relay a second signal through MyD88 that leads to the activation of the transcription factor NF-kappa B. So B cells that are responding to Ti antigens are defined by the activation of AP1, NFAT, and NF-kappa B transcriptional programs. Thymus-independent antigens can be further subdivided into T-independent 1, or Ti1, as well as T-independent 2, or Ti2 antigens, which differ with respect to the types of repetitive epitopes that they bind to, as well as the clonality of their antibody responses. For this introductory course, we won't discuss these subtypes any further, but just know that thymus-independent antigens can come in a couple of different categories. Instead, I want you to focus on a few properties that are shared by both categories of thymus-independent antigens, since these will be important to compare to thymus-dependent antigens. Because Ti antigens do not require interactions between B cells and T cells for antibody production, they don't actually prime any sort of T cell response. Ti antigens stimulate B cell responses during the first phase of primary or initial infection, since responding B cells rapidly are capable of producing antibodies. Mature B cells responding to Ti antigens also first produce these antibodies as the IgM isotype. And this is the same isotype that is just initially expressed by mature naive B cells. Although there is some evidence that these cells can undergo a process called class switching to form other antibody isotypes later on. And lastly, a really important distinction for Ti antigens is that the antibody responses they stimulate consist of immunoglobulin molecules whose specificity remains fixed over time. And this, as we'll come to see in the upcoming slides, is an important difference between some B cell responses to thymus-independent versus dependent antigens. The second category of antigens are called thymus-dependent, or TD antigens, and this is because they require T cell help from T follicular helper, or TFH cells, in order to produce their antibodies. These are antigens that are recognized by the BCR, which in complex with the B cell co-receptor complex trigger RAS and MAP kinase signaling, which induces activation of the transcription factors AP1 and NFAT. At this point, the first transcription factor output of the B cell is just the same as we saw during the first step of thymus-independent antigen responses. However, TD antigens diverge at this point by virtue of the fact that they then internalize their BCR that's bound to cognate antigen. And this allows them to degrade and process the antigen for presentation on MHC class II. B cells therefore act as professional APCs in this context, as they present cognate antigen that is then recognized by CD4 positive TFH cells. Note that the TCR on the TFH cell recognizes the same foreign peptide as the BCR, and this is a phenomenon that is called linked recognition. Activation of the TFH cell leads to the production of signals that stimulate B cell proliferation and survival. And this includes a marker called CD40 ligand that binds to a receptor called CD40 that's expressed on the B cell. CD40 signaling within the B cell stimulates a new signal transduction pathway that we haven't discussed yet, which is a non-canonical NF-kappa B pathway activated through the function of a kinase called Nick and a precursor protein called P100. Importantly, this pathway upregulates expression of a key anti-apoptotic protein called BCL2, and this functions by promoting B cell survival. At this point, we understand that BCR activation and CD40 ligand expression by TFH cells is capable of triggering AP1, NFAT, and non-canonical NF-kappa B activation in those B cells. Additionally, TFH cells produce a few other signals that fall under the umbrella of T cell help. These include a cytokine called IL-21, which activates the transcription factor STAT3 in B cells, and this leads to their proliferation and differentiation towards different types of B cell populations. TFH cells can also secrete a variety of other cytokines related to the specific type of infection taking place, and this can influence the process of class switch recombination that we will cover at the end of this lecture. Collectively, molecules such as CD40 ligand and IL-21 represent what we refer to as B cell help, which are the mechanisms through which TFH cells support B cell activation in response to thymus-dependent antigens. These transcriptional programs induced by T cell help contribute additional signals that promote the activation of B cells in different ways than we see in response to thymus-independent antigens, which only induce AP1, NFAT, and canonical NF-kappa B signaling. B cells that have become activated by T cell help can undergo a few different fates, which may be easier to think about if we map these differentiation steps to their localization within secondary lymphoid organs. Before we learn more about how thymus-dependent antigens influence B cell fates downstream of activation, it's worth considering where these processes occur in the secondary lymphoid organs. In order to increase the likelihood that B cells and T cells whose antigen receptors recognize the same antigen will actually find each other and be able to interact with each other, secondary lymphoid organs coordinate T and B cell localization through a few different sets of chemokines and chemokine receptors. Now, the white pulp of the spleen contains high concentrations of lymphocytes, which are further divided into T cell zones, shown in blue, and B cell zones, or B cell follicles, that are shown here in yellow. T cell zones express high concentrations of the chemokine CCL21, which attracts CCR7 expressing naive T cells and retains them within this region. B cell follicles contain follicular dendritic cells that help secrete the chemokine CXCL13, which attracts CXCR5 expressing naive B cells and retains them within the B cell zone. Follicles also contain high concentrations of two related cytokines that are important for B cell survival and activation, which are called BAFF and APRIL. Now, upon recognition of cognate thymus-dependent antigen, a B cell, shown here in yellow, undergoes an activation signal through its BCR and co-receptor that we just discussed in the last slide. This makes the B cell downregulate expression of CXCR5 and instead upregulate expression of CCR7. These activated B cells also increase expression of a chemokine receptor called EBI2, which recognizes oxysterols such as 7-alpha and 25-HC, which are expressed in the outer and interfollicular regions. Together, CCR7 and EBI2 expression bring B cells to the interface between the follicle and the T cell zone. And this migration process takes about 24 hours following BCR and co-receptor activation. At the same time, the T cells that are shown here in blue, which have undergone TCR signaling and become activated during that same immune response, will actually downregulate expression of CCR7 and upregulate expression of CXCR5. And this brings the activated T cell to that same border area between individual follicles, which is called the interfollicular zone. For T and B cells that have become activated within the same time frame in response to the same infection, there is a higher likelihood that they might recognize the same antigen through linked recognition. So the concentration of activated T and B cells along the border of each of these zones increases the chances of a productive interaction between B cell and TFH cell. And this could lead to the induction of transcriptional programs in B cells that we covered in the last slide. B cells that receive T cell help induce the variety of transcriptional programs that we summarized a couple of slides ago. Some outputs of T cell help include B cell proliferation, as well as the decreased expression of CCR7. This is paired with increased expression of EBI2, which results in B cell migration to the outer follicle as well as the interfollicular regions. The precise localization of these activated B cells and the cell types that they interact with then determine whether these B cells can go down one of two fate-determining pathways. The first of these is when activated B cells no longer interact with T follicular helper cells. So these B cells can either migrate to an area outside of the follicle where they form a cellular aggregate called the primary focus, or they can remain in the follicle. Primary focus B cells differentiate into something called a plasmablast, which is a B cell that rapidly produces large amounts of antibodies. B cells that remain in the follicle without any further T cell interactions differentiate into a type of B cell called a plasma cell. So plasmablasts rapidly proliferate for several days and churn out large amounts of antibodies. And these are really protective during the first phase of the primary B cell response. These cells also maintain expression of surface immunoglobulin as BCRs, while also secreting soluble antibodies. Like B cells responding to Thymus-independent antigens, most of these antibodies are the constitutively expressed IgM isotype, which provides immediate protection. However, plasmablasts are relatively short-lived and will stop proliferating and die within about 10 days. In comparison, plasma cells do not express surface immunoglobulin, but still secrete antibodies and are not as proliferative. However, some of these cells are long-lived and eventually migrate to the bone marrow, where they form a stable pool of memory plasma cells. We will hear more about these in Lecture 5C, where we discuss the formation and reactivation of memory B cells. The second pathway that B cells can go down at this point is one where they remain in the follicle in close association with the T follicular helper cells that provided T cell help. Here, both B cells and TFH cells form something called the Germinal center, or GC, which represents the second phase of the primary B cell response to infection. Lymphoid follicles that contain germinal centers are also called secondary lymphoid follicles. A number of important mechanisms related to the specialization of antibody responses take place in the germinal center, which we will discuss next. And please note that in addition to plasma cells that we just introduced, germinal center B cells can also form long-lived memory B cells. Germinal centers are important structures where the second phase of an initial B cell response is engaged, and this has the overall goal of fine-tuning antibody responses to be the most effective at eliminating a specific type of infection. This goal is accomplished through two mechanisms that we will discuss as we finish today's lecture, and these consist of somatic hypermutation, which enables a process called affinity maturation, as well as class switching of antibody isotypes. These mechanisms allow activated B cells in germinal centers to produce antibodies that are really fully optimized to fight a specific type of infection. And therefore, the germinal center yields higher quality antibodies than those produced by plasmablasts in the primary focus. However, germinal center B cells take longer to optimize these antibodies. Now, germinal centers are specialized microenvironments that are primarily made up of proliferating B cells combined with some antigen-specific TFH cells. And GCs can be divided into three distinct zones that differ with respect to their cellular composition, as well as the biological mechanisms that take place within each area. Since GCs are formed in existing B cell follicles of secondary lymphoid organs, they will push non-activated B cells to the edges of the follicle, and this creates an area called the mantle zone. This contains non-activated and non-proliferating B cells that do not participate in germinal center reactions. The dark zone of the GC is named because it appears darker by microscopy, and this is due to the fact that it contains a high density of rapidly proliferating B cells called centroblasts. These cells express the chemokine receptor CXCR4, which attracts them to the chemokine CXCL12, which is produced in the dark zone. Importantly, the dark zone is the region of the germinal center where the genes encoding immunoglobulin are mutated, and this is again through the process of somatic hypermutation, which we will discuss in the upcoming slides. The light zone is a less densely packed region that contains TFH cells as well as some more slowly proliferating B cells that emerge from the dark zone, and these B cells are called centrocytes. Upon completion of mutation mechanisms in the dark zone, these B cells lose CXCR4 expression but maintain CXCR5 expression, which draws them to CXCL13 that's produced by follicular dendritic cells located in the light zone. Importantly, the light zone is the region of the germinal center where B cells that have mutated immunoglobulin proteins in the dark zone now interact with TFH cells to test their new form of immunoglobulin, and also they can undergo class switching here. Note that B cells in the germinal center can undergo multiple rounds of mutations by cycling between the light and dark zones, and that this cycling is critical for optimizing antibody responses. Let's next learn about the molecular mechanisms underlying the first process of antibody optimization, which is somatic hypermutation. This occurs in the DNA loci encoding the variable region of the immunoglobulin molecule at both V heavy and V light chains. Somatic hypermutation takes place in rapidly proliferating centroblasts found in the dark zone of the germinal center. When the genes for this region are transcribed, they temporarily unwind the DNA helix to form single-stranded DNA. And this single-stranded DNA is then attacked by an enzyme named AID that is only expressed in B cells. AID is a cytidine deaminase, which means that it acts by attacking the amine group on cytidine, and it deaminates this nucleotide, which converts it to uridine, which is also abbreviated as U. Uridine is foreign to normal DNA, and its pairing to guanosine is considered a form of DNA mismatch. So the introduction of uridine by AID triggers multiple forms of DNA repair, including base excision repair as well as mismatch repair. Mismatch repair occurs when the uridine-guanosine pairing is detected by the proteins MSH2 and MSH6, which lead to the excision of the uridine-guanosine pairing as well as several adjacent nucleotides. These excised bases are then filled in with nucleotides by the DNA polymerase Pol-mu, which is error-prone and introduces mutations at A and T base pairs. Secondly, base excision repair occurs when a base excision repair enzyme called UNG removes uridine to create an abasic or empty site in the DNA. This leads to mutations being introduced at C and G base pairs during DNA replication. So taken together, both forms of DNA repair introduce mutations in the variable heavy and variable light chains of immunoglobulin, and we refer to these as somatic hypermutation. The modification of variable light and heavy chains through somatic hypermutation drives a process called affinity maturation, which is best demonstrated by looking at the results of a variable region sequencing experiment. This plot shows individual V region sequences that are represented by horizontal lines, as well as the 3 CDRs of each chain that are shaded in pink, and lastly, amino acid mutations that are indicated by the red rectangles. Sequences are plotted over time as defined by the primary response to infection at 7 days at the top, the secondary response, which is a first immunization that resembles reinfection at 14 days, and lastly, a tertiary response, which is performed after a second immunization at 21 days. Now, as the somatic hypermutation machinery introduces mutations in the variable region of these immunoglobulin loci, B cells acquire amino acid mutations in their CDRs. Those mutations that improve the antibody's affinity for antigen allow that B cell to more efficiently bind antigen, which relays survival signals that allow that B cell clone to outcompete those whose mutations did not confer improved affinity for antigen. This process can be repeated several times as a B cell clone cycles between the dark zone of the GC, where somatic hypermutation occurs, and the light zone, where B cell selection occurs. And this means that antibody affinity is optimized over time. This is represented in the figure by the accumulation of more mutations in the CDRs of a clone's immunoglobulin, which ultimately lead to the end result of generating antibodies with vastly improved affinity to cognate antigen. This entire process is referred to as affinity maturation and is a key component of the germinal center B cell response. Another important aspect of fine-tuning the antibodies produced by B cells is class switch recombination, which allows germinal center B cells to take the IgM or IgD constant regions that are first expressed by activated B cells and swap them out for other constant region segments encoding different antibody isotypes. So if you remember back to our lecture on lymphocyte antigen receptors, the immunoglobulin heavy chain locus also contains numerous constant gene segments that can be paired with a given set of recombined V, D, and J gene segments that make up the variable region. Class switch recombination is a form of non-homologous DNA recombination that only occurs in B cells that have been activated by cognate antigen. The specific constant region gene segment that is selected during class switch recombination depends on the types of cytokines detected by the B cell in the germinal center, though we will discuss how those cytokines correspond with different antibody isotypes in the next lecture. This mechanism is guided by the presence of repetitive stretches of DNA called switch regions, which are located upstream of every constant gene segment. If a B cell receives certain cytokine cues from a TFH cell, the switch region corresponding to a given constant gene segment is transcribed. This typically occurs at 2 switch regions. So first is the one encoding IgM or C-mu, which is the constant region used to make antibodies at baseline. And then the second gene segment is the one that is induced by detected cytokines in the B cell. In this example, this is the isotype IgE encoded by the C epsilon region. However, RNA polymerase, that's shown here in gray, will stall at switch regions due to their repetitive sequences. And this leaves single-stranded DNA vulnerable to attack by the enzyme AID that we just learned about in somatic hypermutation. AID is also involved in class switching since it converts a cytidine to a uridine at this switch region, which is then targeted by the enzymes UNG and APE1, and these work to introduce a high density of single-stranded nicks that ultimately form double-stranded breaks in the DNA. These double-stranded breaks are then targeted by double-stranded break repair machinery, and these include the DNA PK and KU proteins that we learned about during VDJ recombination. This machinery functions by joining 2 transcribed switch regions that were cut by double-stranded breaks and excising all of the intervening sequences. And importantly, this includes excision of the original C-mu gene segment encoding IgM. The remaining DNA is then joined together through DNA ligation. And the end result is that there is a single recombined VDJ combination that's expressed by that B cell clone that is now paired with a new constant gene segment, which in this case is C epsilon, which again encodes IgE. These molecular mechanisms that underlie class switch recombination therefore allow the antibody response to a certain pathogen to be further fine-tuned beyond affinity maturation. We will learn more about the consequences that antibody classes have on humoral immune responses mediated through antibodies in our topics that we cover in the next lecture when we discuss antibody functions in promoting host immunity. We've discussed a lot of molecular mechanisms underlying B cell signaling and activation today, so I just want to end by mapping how each of these processes influence the antibody repertoire of an individual. From our lectures on B cell development and the activation of mature naive B cells, we know that these B cell clones express a specific variable region that is paired with the IgM isotype in its constant region. We know that these antibodies can be produced by B cells responding to Thymus-independent antigens, as well as those responding to Thymus-dependent antigens that no longer interact with TFH cells. So these are either plasmablasts that are found in the primary focus of secondary lymphoid organs, or long-lived plasma cells that originate from the B cell follicle. These IgM antibodies are secreted by the first week of B cell activation in response to infection with a given pathogen. And so these are very important for providing early protection during the first phase of a B cell response. At the end of today's talk, we learned about B cells that, along with TFH cells, establish something called the germinal center that's present in B cell follicles. Now, GC B cells undergo repeated rounds of somatic hypermutation, which introduces mutations in their variable region of the immunoglobulin molecule. If these mutations improve that B cell clone's affinity for antigen, they are selected for over time. Germinal center B cells can also undergo class switch recombination to pair their variable region with a different constant region, and this is said to change the class or isotype of the antibodies that are produced by that clone. Although the germinal center reactions that modify immunoglobulin take a longer period of time and take place over the second phase of the B cell response, they ultimately yield higher quality antibodies that can bind with higher affinity to a given antigen, as well as execute optimized effector functions through their antibody class. We will learn more about these effector functions and how these B cell subsets generate adaptive immune memory in the next lecture. We've covered a lot of material today related to BCR signaling, which thankfully contains a lot of analogous pathways that follow the same logic of TCR signaling that we learned about back in week 4 of this course. We've also been introduced to the different types of antigens that can be recognized by B cells and how they differ with respect to the requirements for T cell help from TFH cells in secondary lymphoid organs. The presence or absence of T cell help has important effects on the transcriptional programs activated within B cells, which then translates to differences in B cell differentiation and the quality of antibodies that they can produce according to the first and second phases of the B cell response. And most of this information is just summarized in these bullet points here. We have one more lecture for this week's B cell module, which is lecture 4C, where we will take what we now know about B cell differentiation and antibody alterations and learn how these things influence the functions of B cells and antibodies during an adaptive immune response.